Today we are going to see the topic regeneration. I am Dr. J. Asnat Mary, Assistant Professor, PG and Research Department of Zoology, Fatima College, Madurai. Regeneration, it can be defined as the natural ability of living organisms to replace worn out parts, repair or renew damaged or lost parts of the body. In simple words, we can say that the replacement of the lost parts of the organism is called regeneration. You must have seen that in lizard the tail is lost, after few weeks it will be regenerated. So likewise there are so many examples, particularly it is very much higher in lower organisms. The regeneration process was first discovered by Trumbull in 1740. The laws of regeneration. The capacity for regeneration is greater in simple organisms, that means compared to vertebrates, in invertebrates, particularly in lower organisms like Hydra, Planaria, it is very much higher and it diminishes with more complex forms in the animal kingdom. So, as the organisms evolved in the phylogenetic, in the evolutionary process, the regeneration capacity is also getting slowly reduced or diminished. The ability for regeneration may be greater in the earlier stages of development. Here is the detail of regeneration in invertebrates. The ability for regeneration is higher in protozoans. If an amoeba is cut into 2 or more pieces, every nucleated piece grows into a complex amoeba. The same phenomenon is also observed in ciliates like Spirostomum, etc. The ability for regeneration is remarkable in coelentrates. If a hydra is cut into 2 pieces, the anterior piece develops a basal disc at the cut end and the posterior piece develops the mouth and the tentacles at the cut end. Even if a hydra is cut into 200 pieces, each piece reconstitutes into a whole miniature hydra. Among platyhelminthes, the process of regeneration is exceedingly higher in Planaria. Any small piece of planarian worm develops into a whole animal, just like in hydra. In annelids, the regeneration capacity is exhibited by polychaetes and aliochaetes. If the worm is cut into 2 pieces, the anterior piece develops a posterior region and the posterior piece regenerates a head region. These 2 individuals are produced. Among annelids, regeneration is somewhat higher in Chaetopteris. The complete individual can be regenerated from any one of the anterior 14 segments. In arthropods, the regeneration is restricted to the replacement of the lost appendages. In case of vertebrates, but in fishes, the ability for regeneration is restricted only to the replacement of fins. The capacity for regeneration is most astonishing in amphibians. Urodeles like newts and salamanders, limbs, tails, external gills, jaws and even part of eye can be regenerated. Among reptiles, the regeneration is restricted to the replacement of tails in lizards. In birds, feathers and parts of the beak can be regenerated. In mammals, the regeneration process is very poor. It includes the replacement of nails, antlers, skin, erythrocytes, internal lining of gut and uterus and so on. The next topic is types of regeneration. Animals exhibits 4 types of regeneration. They are morphallaxis. This type of regeneration is exhibited by lower animals. If a hydra or a planaria is cut into 2 or more pieces, each piece will reconstitute itself into a new and complete individual of a diminished size. Even a small part of the original animal can regenerate a complex whole. This process of reorganization of a small piece into a whole animal is called morphallaxis or morphallactic regeneration. The second one is epimorphosis. This type of regeneration depends upon the growth of new and properly patterned structures. This is the replacement of the lost parts or repair of the damaged body organs. In this type of regeneration, wound is repaired or closed by the expansion of the adjoining epidermis over the wound. For example, in salamanders, if the limb is amputated, it can be regenerated within a few weeks. In these animals, the regenerated limb is an addition to the other parts. Such type of regeneration is called epimorphosis or epimorphic regeneration. Example: regeneration of limbs in salamanders, regeneration of lost tail in lizard, healing of wound and replacement of damaged cells. And then third type of regeneration is called autonomy. In some animals like starfish, some part of the body is broken off on being threatened by a predator. This phenomenon is of self-mutilation of the body is called autonomy. For example, crabs break off their leg on approaching of the enemy, holotherians throw off their internal viscera, and echinoderms like starfish and brittle star sea lily. The fourth type is called heteromorphosis, where the regeneration is faster in the younger than in the adult. The regenerated part may not be always similar to the last part. This type of regeneration is called heteromorphosis. When a particular organ is amputated, the remaining portion in some rare cases may develop into a different organ. This process is called heteromorphosis and heteromorphic regeneration. For example, in the shrimp, the eye may regenerate after being removed. If the eye alone is removed, it will regenerate soon. If the eye is removed along with the optic ganglion, the eye will not be regenerated. Instead of the lost eye, an antenna-like organ develops. This forms a typical case of heteromorphosis. So, you can follow in this picture where there is an amputation of eye. The eye is removed, but in place of eye there is a development of antenna. So, this is an example for heteromorphosis. So, the next is events in regeneration. Among vertebrates, the regenerative capacity is most astonishing in the urodile amphibians like newts and salamanders. The limbs, tails, gills or even jaw of these animals regenerate into remarkable manner both in the larval and adult stages. So, it involves various events. Number 1, wound closure. This is the first event in the regeneration. When there is a wound, there is a bleeding. The blood clots within 30 minutes on the surface of the wound. It prevents the further loss of blood and the direct exposure of the wound to the external environment. Then the epidermal cells migrate all sides towards the center of the wound. Thus, the wound is completely covered with epidermis. This event occurs within a day. Next, the second step is demolition and defense. The next event is the release of toxic substances by the injured tissues. The damaged tissues and debris are removed by autolysis and phagocytosis. This is favored by the increased dilation of blood vessels. And the third step is dedifferentiation. The process of dedifferentiation refers to the reversion of the differentiated cells to totipotent cells. The cells from adjacent epidermis, muscles, nerves, cartilage, connective tissue, etc. undergo dedifferentiation. That means the highly differentiated cells like muscle cells, nerve cells, the transformed into an embryonic cells which has the totipotency for the regenerative process. When sufficient number of totipotent cells are produced, de-differentiation stops. Then the fourth step is blastema formation. The next event in regeneration is the formation of blastema or regeneration bud. De-differentiated totipotent cells begin to accumulate beneath the epidermis on the surface of the wound. As more and more cells aggregate below the epidermis is pushed out and a conical projection appears. This is called blastema or regeneration bud. It is formed of an outer cap-like epidermis and the central core of de-differentiated cells. And the next step is growth. The blastema then grows and increases in size. The growth is caused by the mitotic divisions of the differentiated cells. The last and the final step is differentiation. The blastema differentiates to replace the last parts. The blastema of the forearm differentiate into muscle, bone, cartilage, skin, digits, etc. The blastema differentiates exactly like a limb bud in the embryo.